Hello, my lovely anatomists and physiologists. Welcome back. We are ready to tell the story of our immune response to one of our infected self cells. So let us draw a cell over here. It's just kind of like a circle. And then let's just make it cute. So I'm going to do like a little face for myself. And so what we're going to see is our very first step that's happening. Let me actually not number this yet. Let's say we get infection. So the self cell becomes infected. So we're talking about a pathogen such as a bacterium, which is a singular word for bacteria, it's plural, or let's say a virus infects our cell. And so the very first thing that's happening to initiate our immune response is what is called antigen presentation. Remember, antigen presentation is where our cell that's infected takes an antigen from that pathogen, incorporates it into its major histocompatibility complex protein that it's making all the time to hang off of its cell membrane. And so you end up with, in this case, MHC class 1 is sort of holding out the antigen from the pathogen. So now I'm going to change my smiley face to like a frowny face because my cell's infected. And I'm going to create a little flag and I'm going to make it be sort of like triangular so I have a shape. And we will indicate here that our triangular shape is our antigen. So let's zoom in super close and let's label this as our antigen, our triangle shape. Oops. Okay. So thinking about our pattern, we said the very first step in initiating any adaptive immune response is antigen presentation. And the very second step is an inactive lymphocyte recognizes and binds. So here, remember, we are looking at class 1 MHC markers. And so what that means is it will be a CD8 T cell that will recognize and bind. And we can talk about this as clonal selection, meaning we have, remember, in our bodies, 100 trillion different types of T cells, meaning we have 100 trillion different types of receptors for all the different possible antigens that we might come across in our life. And so when that CD8 T cell is recognizing and binding, what that's doing is actually selecting for that particular one out of the 100 trillion different types of T cells, right? And so let's draw in our CD8 T cell. So I'm going to just do a circle. And this is sort of, I'm just going to say a badass cell. So I'm going to give it some sunglasses, you know, because that automatically means, ooh, we are cool, we are serious. It's just kind of fun. And this cell is so cool, it doesn't even, it just has like a little line for a mouth. This is our CD8 T cell. And what it's going to do is recognize and bind. So I'm going to draw out sort of like my receptor coming off, but just to kind of show the specificity here, I'm going to try to represent my receptor as having a shape specific toward that triangle. So we're getting recognition and binding. We're getting a high level of specificity here, right? And so then our next step, which is not described, I didn't see it described in our OpenStax textbook, but I've read it in other resources, and it's a step we do need to know, is called co-stimulation. And we should think about co-stimulation as the go-ahead. So in this case, ultimately, the CD8 T cell, when it recognizes and binds, when it's activated, it's going to actually lead to the destruction of this infected self cell. And so we don't want this to just randomly happen. We want to be very sure about it. And so we can consider that the infected cell gives this go ahead, like, yes, I'll take one for the team. Because remember, ultimately this cell will die, but this is for like the greater good of the whole body, right? So this is killing not just the cell, but also the pathogen that's infected it. And so that should preserve other cells in the body. So we're ready for our last step, which is the CD8 T cell will become active. So we can say activates. And what this actually means is it will divide and differentiate. And so when it divides, that means we're getting we're getting cell division, right? We're getting more cells. And in this case, we're getting 3 types of cells. So the CD8 T cell will divide and differentiate, forming a memory T cell, a regulatory T cell, and a cytotoxic T cell. Okay, so when our CD8 T cell divides and differentiates, we get these 3 possible cells. We get the memory T cell, regulatory T cell, excuse me, and the cytotoxic T cell. We can abbreviate the cytotoxic T cell with a big T, little c, and then we can call it a cell. So let me just leave it without abbreviation. The cytotoxic T cell is the cell that is going to be functioning here in this primary immune response. So primary immune response means, again, this is our first time we're ever exposed. This is the first time we're ever exposed. What are we going to do? These are the steps of what we're going to do. When we get that cytotoxic T cell, what we see happening is it will actually kill the target cell. And it's going to do this basically by inducing apoptosis, just like the NK cell when we looked at immunological surveillance. So remember, that means the NK cell was releasing the perforin to poke holes in the membrane of this cell, and then also the granzyme to digest that cell. Or maybe it's releasing the fast ligand, which is going to trigger apoptosis in this target cell. And then what we can see happening is it can kill more than 1 cell at a time. This is why we call the cytotoxic T cell like the cell-mediated combat, the cell-mediated defense, the cell-to-cell defense. It's gonna actually, let me do this in red. The cytotoxic T cell is actually going to be the cell that then is poking holes in the membrane, right? So let's poke some holes in there. And just like, you know, if you have a bunch of holes in your body and your insides leak out, that's bad. If you poke a bunch of holes in a cell, the insides leak out and that's bad. Or, you know, we can talk about those other chemicals going into the cell in order to digest it from the inside. And so ultimately we see that this cell will die. We see that other infected cells will die. And so the cytotoxic T cell is this really good cell that's toxic to cells. It's going to go around, find those that are infected with this particular antigen and destroy them. Okay. It took a little bit of time to get to that particular step. So that's why the first primary immune response, the first time you're exposed to a disease, to pathogen, you are sick with that particular pathogen. The memory cell we see is going to be the cell that's actually long-lived. This is the beautiful part of your adaptive immune response. So you make these memory T cells that can be present for the rest of your life. So you can be exposed to this pathogen at 3 months old, and then you have immunity. You have a memory cell in place in your body for the rest of your days, okay? This memory cell is going to be able to act very fast as part of your secondary immune response. Meaning, the next time you're exposed to this particular pathogen, we're right away taking action to destroy those cells, such that very few of your cells get infected and very few of your cells have to be destroyed, which means, you know, you're not even aware that this is happening inside of your body because you have no symptoms. Or, you know, if it is something where you do have some symptoms, it's way lighter than what it was that first go round. Okay. Now keep in mind that this whole immune response is generating these cytotoxic T cells, these cells that go around and actively kill your cells. And so we don't want this to be a response that gets way out of control. And so that is the job of the regulatory T cell. So we can also talk about it as a suppressor cell. It's going to suppress our T cell responses. Remember, this is a pattern we see in our body where we have these control mechanisms in place. So we have this ability to generate these cytotoxic T cells and destroy the cells of our body, but we want to add a level of control to that. That's the job of the regulatory T cell. And, you know, when it's working great, it prevents autoimmune response. An autoimmune response is when your immune system does attack your cells. And there are several examples of that that are given to us in our textbook if you'd like to read a little bit. Okay, just double checking my notes. This is a pretty good overview of this process. Stay tuned. Up next, we want to look at what happens when a professional antigen presenting cell initiates or is triggering the immune response. So stay tuned. As always, take care of yourselves and each other.